Hello, everyone, and welcome to People Behind the Science. I'm your host, Dr. Marie McNeely, and I appreciate you joining me today for episode 687 with our guest, Dr. Michele Battle. Now, listeners, if you like this episode and you want to check out more stories from amazing people who are doing great science today, you can find more episodes of our show on Apple Podcasts, Google Podcasts, Spotify, or wherever else you like to listen. And today, Michele has some excellent stories to share with all of us. So listeners, get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Michele Battle. So, Michele, welcome to our show today. How are you?
Thank you so much. I'm great. It's very exciting to be here.
Well, we are thrilled to have you with us, and we're looking forward to learning from you today and learning more about you today. So let me first start by telling our listeners a little bit more about your background. So listeners, Michele is an associate professor of cell biology, neurobiology, and anatomy at the Medical College of Wisconsin. She received her bachelor's degree in biology and philosophy from the University of Scranton and her PhD in cell and molecular biology from Michigan State University. And before joining the faculty, she conducted postdoctoral research at the Medical College of Wisconsin, and she was the recipient of the inaugural Edward J. Lennon, MD Award for an Outstanding Women Postdoctoral Researcher. Michele has also been awarded the Research Scholar Award from the American Gastroenterological Association Research Foundation, the Presidential Scholar Award from the Medical College of Wisconsin, the Women in Science Rising Pioneer Award from the Medical College of Wisconsin, and also numerous awards from the Medical College of Wisconsin in recognition of her excellence in teaching. And in addition, she was named a Fellow of the Kavli Foundation in 2015. And today, Michele, we're excited to get to know you both as a scientist, but also more generally as a person. So can you start by telling us what you like to do when you're not doing science?
I'm a very people-oriented person, so my favorite thing to do really is spend time with my family and friends. It doesn't particularly matter what we're doing. I just like to be with them and make those connections. With my family, we've recently added a puppy, so we spend a lot of time walking the puppy and going to the dog park. It's really fun living near Lake Michigan to be able to walk by the lake, run by the lake, just sit by the lake and sort of meditate on it. We do a lot of hiking, and my kids and I have gotten into a little bit of ropes adventure courses. So we like to get out there and get active. But for the most part, my favorite thing to do is really just be with my family and friends and experience them and spend time with them.
Oh, that sounds absolutely wonderful. Congratulations on the new puppy being added to your family. What kind of dog is it?
She is a rescue dog. She came from a shelter in Illinois. She had been rescued from a hoarder's house in Texas. And she is a part Labrador, part Pit Bull, part Chow Chow. And then, of course, a scientist. You know, we did the DNA test. So she's also part Super Mutt, which is my favorite part, which is a mix of many breeds. She's full of energy and she keeps us on our toes, but it's been fantastic to have that companionship. And I really never thought I'd have a dog, but bringing the dog into the family has been very energizing to us. And my daughter, knowing how to appeal to a person of reason and science, made a PowerPoint presentation.
Oh, wow.
To convince my husband and I about all of the fantastic pros of adding a dog to our family.
Very persuasive. I like it. And it sounds like she won out in the end.
She did.
Well, it sounds like so much fun there, Michele. I'm glad to hear that you have all these different activities that you like to do with family and friends to kind of take a break from the science, because I think that is super important. But I'd love to chat about your scientific side as well. You are doing amazing work there in your lab. So how do you describe your research to people who aren't familiar with your particular research area?
Basically, I tell people that I study the gut, and we do that because we want to understand how the organs in the gut form. Because if we know how these organs form and function in normal health, we can apply that to diseases. So we're really looking at the basic mechanisms that allow us to have a functional gut, so that we can then figure out how to fix it when things go wrong.
I think that is a great description. And it's something you sort of bring it back to something everyone can relate to. I think one of the challenges scientists run into sometimes in talking about their work is they kind of go into the detail immediately. But I think starting with that kind of more general, big picture view can really help people connect with and relate to your work.
Oh, absolutely. I think as scientists too, we have a language and a jargon that can be very unapproachable. So always remembering to step back from that, I think is important so that you can clearly communicate and not get stuck in our language.
Absolutely. And I love hearing a little bit more about what motivates scientists. I know it's a struggle sometimes in the lab and the research that you're doing, but having a little quote or a saying or a mantra in your mind can keep you going when times get tough. So Michelle, do you have a favorite quote or just a saying that really resonates with you?
I'm glad you said the word mantra because I think of this as sort of a mantra of confidence. And it actually comes from a professor of mine back in undergraduate education. Professor Klonowski was one of my philosophy professors. And in a time when I was lacking in confidence in philosophy, particularly, I wasn't sure that I really had the mind for philosophy. He wrote on one of my exams, which I still have, and I hang it up, "Be the woman." And to me, that really encapsulated everything about being a confident woman. Really be the woman, be who you are. Don't be afraid to be who you are. Go for what you want. Go out and get it. And so whenever I have these moments of self-doubt, I go back to that mantra of confidence, be the woman. And it really empowers me and inspires me to move forward and to tackle hard things and to work through the struggles that we all face in life and whatever we do, science and otherwise.
Definitely. I love that mantra. And it sounds like this philosophy teacher's comment, maybe they didn't even think about it much at the moment when they wrote that on your paper, but it really stuck with you. And I think that is the case a lot of times with these people who have shaped our lives or had a big impact. So are there others that really rise to the top of your mind, Michelle, in terms of mentors or role models or people who really shaped your future?
I can't think of role models without thinking of one of my co-mentors for my PhD, Dr. Veronica Maher. Veronica was really a force of nature. The things I admired about her or the ways that she inspired me really came from how she came up in science. It was surprising to me to learn that my university distinguished professor at Michigan State University was not only a phenomenal scientist, but she had taken on another role in her life, and she was actually a Catholic nun. It surprised me a bit being at a state school and finding this out. But when I talked to her then more about what it was like for her in the 1960s to be studying a PhD, she not only was one of the only women were not common in the field, but she also had this extra struggle of being a pre-Vatican II nun in full habit working in the lab.
Wow.
She had a lot of interesting stares. There were a lot of questions about how serious she was. What was she going to do with this PhD? Would she really be a successful scientist? Is it worth our time to educate this woman? And she proved them all that she was worth it. She went on to do a postdoc at Yale University. She went on to establish the carcinogenesis lab at Michigan State. As I said, she was a university distinguished professor. She published fantastic work. She trained many, many, many scientists, including myself. And she really let me realize that you can't let the expectations of others define you. You have to define yourself. And you can't let what others think really define you. You need to show them who you are and not be afraid. And so she really showed me confidence, persistence, tenacity, and just even to today, she's passed away now, but even to today, I think about her often. I often wish she was around so I could tell her about various successes we've had in the lab, successes with my students, and even successes in my family and things going on in my family. She was really just a spectacular woman.
That sounds like a phenomenal role model. And I really love that you really described how she showed you that you can have these different sides of you. You can be interested in science and religion, or you could be a creative person and a scientist. You can have all these different facets of your personality. You don't have to fit the mold of what people expect you to be as just a scientist. And I think that's absolutely wonderful. So, Michelle, if we take a look at your own timeline, can you rewind back to the beginning and tell us a little bit more about how you got interested in science initially? I think everyone has a little bit of a different story in terms of how they got on their own particular career path. So what's your story?
It goes back again to high school. It goes back to my biology teacher, Mrs. Lotney. who inspired in me a complete fascination with DNA. I remember learning about this as a sophomore in high school and thinking to myself, this is a fantastic molecule. In some ways, it's so simple yet complex. It defines all of us, every species. This same biomolecule, just in different forms and functions, makes a fruit fly, makes a person, makes a mouse. So when I went into college, I wasn't sure I was going to study biology, but it was in the back of my head that biology was one of the fields I wanted to pursue. And then in our freshman biology lab, we did a DNA extraction from the salivary glands of Drosophila. And when I actually literally could spool that DNA out, again, I just was captivated by DNA. And I thought, I've got to understand this molecule. I've got to understand how this works and how the DNA, obviously, we all have the same DNA in all of the cells of our body, but every cell is different. Every cell has its own function. So how does this DNA operate in various cells to make, say, a stomach cell versus a brain cell versus a heart cell? So really that fascination with DNA and how it works led me to want to pursue a PhD in cell and molecular biology at Michigan State University. While I was there, I studied cancer and did this mostly with cells that we would culture in the lab. And that was really exciting to me. But when I moved on to my postdoctoral studies, I wanted to expand my horizons and look to use models other than just cells in culture in the lab. And so that led me to the Medical College of Wisconsin and the lab of Stephen Duncan, where I entered the field of developmental biology. I remember one of my first days in the lab meeting, having never seen a mouse embryo or anything related to developmental biology and thinking to myself, oh my goodness, how am I going to do this? I don't know anything about this. But with great mentorship of Dr. Duncan and with hard work and studying up on things, I really realized that developmental biology was a great way to have a window into disease. Because a lot of things that happen in disease, we can trace back to developmental pathways that either turn on when they shouldn't or get activated in abnormal ways. So I was able then to take what I learned from cancer biology as a graduate student and developmental biology as a postdoctoral fellow and put those together to now study what I do in my lab, which is development of the gut, trying to understand how the cells of the gut take on specific functions. So how does the stomach have an acid-producing cell where the intestine has a cell to absorb nutrients? And if we understand, again, those basic developmental biology pathways that make a cell into its proper identity, then we can understand later on when something goes wrong, how we might be able to identify that earlier, how we might be able to fix that, to come up with a new therapy for a disease. And we're applying that again to cancer research right now, looking at esophageal cancer and gastric cancer.
Absolutely. And it sounds like you had a phenomenal experience there as a postdoc that ultimately led you to stay at the Medical College of Wisconsin. So what was that transition like for you?
It wasn't really the norm at the time. I think it's become more common for people to stay in institutions where they've done training. It was really fantastic for me. I do remember there was a search going on, and I actually never thought to apply for it because it didn't seem to me that people generally stayed in their training spaces to become faculty. But the chair of my department at the time, Dr. Joe Bisharis, approached me and asked if I wanted to apply for the position. And I very calmly said yes. And then I went basically and did cartwheels in the lab because I truly loved and still love being at the Medical College of Wisconsin. The cell biology department in particular was just a very nurturing place, a very collegial place. We were all a big team. We were all working for each other's successes. When one person succeeded, we all succeeded. So it was really fantastic for me to be able to establish my lab in such a supportive environment and also to know the system. When you're starting your lab, there are a lot of things you need to do. There are a lot of basic setup logistics. There are approvals you need to get. And knowing the people and the places to do all of that really made for a seamless transition. I was worried a bit, perhaps, that people might not see me in this new role. So being a postdoctoral fellow and transitioning to faculty, would the people around the medical college recognize that and see that and respect that? And that was a clearly unfounded fear. I had no trouble fitting in as a faculty member. I had the respect of my colleagues and the support of my colleagues. And I think that really being in that supportive environment led me to be able to succeed in getting my lab funded within the first year to year and a half of setting up the lab. So I don't think that I would have been able to do that without the fantastic support of my colleagues. And again, Steve Duncan, who was the best postdoctoral mentor anyone could ask for. He was always looking out for me, looking out for my best interests. Right from day one, he asked, what is your goal? What do you want to get out of your career? And when I said, I want to run a lab, I want to be a PI, we started on day one strategizing for that. And he was very generous in providing me an opportunity to be creative and independent and develop this line of GI research that I would be able to use to establish my own lab.
That sounds wonderful. And I'm really glad you brought up this point of having this fear in the back of your mind that people would still think of you as a trainee. That's something I experienced as well, where I joined the faculty at the same place where I did my graduate education, my PhD. And it was the same thing where I was coming in on day one thinking, are they still going to be looking at me as that graduate student? And I had the same experience you're describing where you really do transition into that role and people do see and respect you as a faculty member. So for our listeners out there who may have similar concerns, it'll be okay.
That's right. It will be. It will be.
Definitely. So you've got some great research going on now in your lab. I'd love to talk about it a little bit more. So Michele, do you have a particular project that you're working on right now that you're really excited about at the moment?
Yes, we just recently got funded by the NIH for a study related to so stomach development and gastric cancer. I'm super excited about that project. It was developed really by a postdoctoral fellow in my lab. She's now moved on to an industry position, which she is doing fantastic in. And in that study, we're really focused on trying to understand, again, the very foundational and basic processes that make gastric cells function normally so that we can understand when things go awry. And so one of the key cells involved in normal gastric function, I think everyone could guess, is the parietal cell, the acid-producing cell. So in order to maintain a healthy stomach, we need to maintain healthy acid secretion, the correct pH. One of the really essential steps in the progression of gastric disease, metaplasia, and cancer is the loss of those cells, the loss of the parietal cells. So we want to understand what makes those parietal cells tick. So we are really interested, again, in transcription factors. So going back to my love of DNA, I want to understand how DNA is read in a given cell. So that happens mainly through the transcription factors that are present and how those transcription factors control which elements of DNA are expressed. So we're looking at this transcription factor called GATA4, and we're looking at its role in defining these parietal cells. And we have some very exciting data that suggests that this protein is essential in making a functional parietal cell. So we're using mouse models and cell models to look at how this protein functions in the parietal cells. And some of our really exciting mouse data shows that when we do get rid of GATA4 selectively in these cells, so we can eliminate it selectively only in the acid-producing cells of the stomach, we see the starts of gastric disease. We start to see these first steps in the progression toward cancer. So right now, we are very excited to dig into that more, to dig into the molecular mechanisms, and also to look at ways we might be able to use this knowledge to either protect the parietal cell population. So are there ways that we can prevent these cells from dying out during disease process? Or can we find ways to regenerate those in stomachs that have started down this road to disease?
Oh, this is fascinating, Michele. I love this project, and I think it's really interesting that you've got kind of these different angles that you might be able to remedy the situation, whether it's sort of the regeneration side of things or the rescue. I think that is absolutely amazing. But I know you encounter challenges in science. Not every day is full of great new data and exciting discoveries. And we love to talk about the challenges as well, because I think oftentimes people only see the finished product in science, whether it's the paper or the press release. So do you have an example of something that you've really struggled with, or perhaps a major failure that you've had, Michele, that you can share with us today?
Yeah, I think every person in science can relate to that. And I always tell my students and trainees that science is persistence. Mm-hmm. And also you have to know that you will fail and you have to be okay with that. So One of the biggest struggles I had came at sort of the mid-career point. When I was promoted to associate professor, I needed to renew my R01 grant. At that time, the startup funds that I had to establish my lab, I had pretty much run through. And so it was absolutely essential to renew this grant. And it was not easy. Mm-hmm. Funding rates at NIH, as we all know, are challenging. And I put in my renewal grant and it didn't get discussed.
Oh, no.
And that was a very terrible feeling. And I remember literally sitting at my computer and crying. And I was crying because I started to doubt myself. I started to doubt that, did I belong here? Was my science worth studying? Was I really in this position because I deserved it? I started to feel that imposter syndrome like I had never felt it before. I tried to then pick myself up and write it again. And I wrote it again and it got a score, but it wasn't fundable. And so at that point, I thought, okay, I've got to be creative here. I've got to find a way forward. And so I learned from that I need to ask people for help, that I wasn't in this sort of fight or battle for this R01 on my own, but I could get help. And so I went to a colleague that I didn't know before. I just reached out to her on a cold email, who was an expert in this area that I was pursuing. And I asked her if she would mentor me, and she would mentor me in this writing of this grant. And I shouldn't have been surprised because I found most of science to be very helpful and creative and collaborative. She was immediately willing to help out. So we had a lot of strategizing meetings, and she helped me to reshape my goals and reshape my aims. And on the fourth try, finally, the grant was scored and it was funded, and it had a 2 percentile, which made me literally scream for joy in my office. People thought something was wrong, but everything was right. So from that experience, I learned that I had to really trust myself. I had to silence those negative voices in my head that were saying, you didn't get this grant because you are not smart enough, you're not creative enough, your science isn't exciting enough. And I had to ask for help and be willing to change, be willing to be flexible, and in that way, move myself forward. And so that was a time where the stress and anxiety was very high. I learned from that I will never let myself feel that way again. And I've learned that I am a worthy scientist and my work is worth it. And that if it doesn't get funded, it's probably just because I need to re-explain it or reshape it. And that the road that we think we're going on to that grant or that goal might not be exactly the road we take. There are many paths to the same destination. And so you have to be willing to change it up and switch it up to get to that final goal or success.
Absolutely. And I think this is such an important but a difficult lesson, particularly for early career scientists, that you do have to do a lot of the hard work yourself, but you don't have to do everything alone. And I think being able to just talk through your ideas, to get feedback and input from someone else, can make all of the difference in one of these grant submissions or just a paper or something that you're working on and really struggling with.
Sometimes the way you're thinking about something is so clear to you, but it means absolutely the opposite to someone else.
Right.
You don't see that until you step out of your comfort zone and ask for real constructive criticism. And it might be hard to listen to. I think we all think of our brands, our ideas as our sort of like our babies. So you're very emotionally attached to them because they're really an integral part of you and your mind, your thought process, your creative process. And so when someone points out something that might be a flaw, sometimes it's hard to hear, but it's absolutely essential to hear. And that's where resilience really comes in, being able to take that constructive comment and criticism and use it to reshape or reframe your ideas and not get caught up in that sort of emotional attachment that we all have to our ideas.
Definitely. And Michelle, I'm thrilled to hear that this story had a happy ending for you. Congratulations, of course, on getting that grant. But I know success in science comes in many forms, and it can be a long time in between successes. So celebrating these little wins along the way can be really important. So do you have a favorite success, whether it was a big win or even a small victory that you'd like to share with us today?
I immediately think back to when I was pursuing my PhD, and probably students out there listening or trainees or maybe even faculty can relate to that grind of working towards something and seeing just failure.
Mm-hmm.
And you're ready to give up almost. And a simple Western blot was my salvation.
Really?
I remember thinking that I was never going to get this degree. Nothing was going to work. And I was doing this experiment to show that these two proteins interacted with each other. And I did the immunoprecipitation experiment and I put the film on the Western blot because back in those days we were still using film. And I left because I thought to myself, this is not going to work. And I came in the next day and developed the film, not thinking much of anything. And lo and behold, the band was there, which told me the proteins interacted.
Oh, wow.
Again, I sat at my desk and cried. I cry a lot, happy tears and sad tears. And my colleague who knew I was going through this really sort of stressful time came over and said, are you okay? It's okay. Everything's okay. And I said, I'm going to graduate. I'm going to have a thesis. And I made a photocopy of that film and I hung it on my refrigerator to remind me that you persevere and you go through these frustrating times, but if you keep working, something good will come out of it. And so that was some might say a very small success, one experiment working, but that experiment working really transformed my thinking, transformed my attitude, and reinvigorated me and re-energized me to know that I could succeed. I would succeed. I would get this degree. And it was just really empowering, just a small Western blot that changed my life in some ways because I was ready to give up.
Absolutely. And I think sometimes that's all you need to help give you that momentum back to get moving in the right direction. So this is a fantastic success story. And again, I think so important to celebrate those little wins because they can make all the difference for you, for your students, and for your research.
Yeah, I tell my students that all the time. Sometimes they are more shy about celebrating something. And I'm like, nope. You got to celebrate this. We celebrate all the wins. You celebrate the wins because that's your fuel. That's going to fuel you through the tough times, the frustrating times.
So how does your lab celebrate the wins?
We do a lot of eating together. We usually go out for lunch or go out to have a little coffee break or something like that together. We always celebrate at lab meeting. We bring up positive wins. I try to remember to start the lab meeting by asking, what are their wins, big or small? What are they looking forward to in the week ahead so that we can start on a positive note? So our celebrations aren't grand in any way, but I make sure that they know that I recognize their wins and that I appreciate them and that we are a team and nothing succeeds without them. The success is all of us working together. And so, like I said, we don't have these major celebrations. We just have these small things, but we try to recognize successes in the smallest form so that we can keep up that positive energy.
Definitely. And I think even these small moments of gratitude and acknowledgement really matter. And like we said, can really keep people going because I think there is so much to do in science. Things can get quite busy. And I know you spend a lot of time reading the literature, reading through your grant applications and writing and things like that. But we like to encourage people to give their minds a break, to pick up something for fun to read. So Michele, do you have a favorite book that you can recommend for our listeners who are looking for something to maybe distract themselves from their own science?
That's a little bit of a challenging question for me. I'm going to tell you why. I have a memory that is great for science. I could tell you that I did this experiment in 2007, and you can find it on page 33 of my lab notebook. But could I tell you the titles and authors of the books I've read and what they're about? No. I have this interesting brain that as soon as I read something, it's out of my brain. So thinking back to some books I've read that I've loved, I can't really tell you much about the plots because I don't remember them now. I'd have to go reread them, which is exciting because it means everything's always new to me. Mm-hmm. But I really enjoyed the author Graham Greene. I took a course actually on Graham Greene back in college, and I've pretty much read all of Graham Greene's works. And one that I do remember standing out was A Burnt Out Case. That was a particularly great novel that I read. But I feel kind of silly that I've read it, loved it, but I could not tell you about it.
No worries.
It's just so strange, a strange personality quirk that I could read it today and it'd be like I never read it before.
Well, I'll admit, I've had those moments where you're like a third of the way into a book and you have that, have I read this before moment? And it turns out, yes, you have.
Yeah, it's one of the quirky things about my mind.
Well, thank you so much for that recommendation. We will add Graham Greene to our list for our listeners to find on our website if they're looking for a great author to pick up. And we've talked about some different aspects of your career. One of the things we haven't yet touched on, Michelle, are some of the opportunities that you get as a scientist to travel, to see the world, to meet with colleagues, to go to conferences. So do you have a favorite place that your science has taken you?
I was fortunate to be able to go to a conference back at the end of my postdoctoral training in Capri, Italy. It was really one of the most beautiful places I've ever been. And it was a fantastic conference, not only because of the setting, which I do think fantastic settings, relaxing settings really help for the science. Being in that kind of frame of mind just helps you think more clearly. But it was a conference that was all about GATA factors. So it was very focused. It was very topic specific. So it was a small group of all the world's experts. And it was the first time I was invited to give a talk at a major conference. So it stands out to me for being not only a fantastic, beautiful place to visit, but the first time I was able to actually showcase my work in an international meeting. And it really was a great time to network with this community of researchers. I liked this meeting too, because there was a lot of time for meals together and socializing outside of the science. And so again, getting to know people and not just their science is so helpful. So I really like those sorts of meetings, the smaller meetings where you really get to talk and socialize about science, about life. More recently, I was fortunate enough to be elected to organize a FASA research conference focused on gastrointestinal biology. It was the 20th iteration of this meeting. It's really a popular meeting. It's considered by many to be the preeminent GI meeting. So I was so honored to be able to organize it. And that's another one of these meetings where it happens in Colorado, in Steamboat Springs. So again, a beautiful, relaxing setting. And it brings together the GI community and allows for that interaction at a scientific level and then also in a social networking way. And there's a huge focus on trainees at that meeting too. So I was really excited that we could offer trainees a lot of opportunities to speak and really promote themselves as they're moving forward through graduate school, into postdocs, into faculty positions. And so in the end, the meetings like that are the ones that really stand out to me, the ones that bring together smaller groups of people in a setting that allows you to relax, enjoy nature, and also then enjoy the science and the people.
Michelle, these both sound like marvelous conference opportunities. And I have to ask, the meeting in Capri, was that your first time in Italy?
It was. It was just fantastic. Capri is just phenomenally beautiful, and the food was fantastic as well.
So if our listeners are able to escape and make it to Capri, what is something they definitely have to check out when they're there?
Oh, you have to take the boat tours that take you around the island because there are fantastic caverns that you can boat into that are just phenomenally beautiful. And then also just hiking around the island is actually fantastic as well. The views are just amazing. And the water is so blue and clear and just pristine.
It sounds absolutely lovely. Thank you so much for telling us more about these wonderful travel experiences that you've had for science. And I think a lot of people have stereotypes about what life is like as a scientist and what scientists themselves are like. And I know research is quite important, but I know so many funny, creative, amazing people who really don't fit the mold of the stereotypical scientists. And I think every lab has quirks and traditions and just personalities. So for you, Michelle, do you have a particular example of one of these human moments in science or a quirk or a tradition that you experienced that goes against some of the stereotypes that people may have?
The most recent thing I can think of that was a sort of funny experience I had was, again, at that FASA meeting. The GI community is fantastic, and there are a group of faculty that have a band, and the band is called GI Distress, which in and of itself is fantastic name for the band. Mm-hmm. So one of the fun things they do is when they play the shows, and they play shows at many conferences, they have a night of open mic. And so that is really fun to experience because you get to see the people at the conference, both the faculty and the trainees, put aside their nervousness and their sort of sense of potentially awkwardness and fear and get up and sing a song and just put it all out there in a song. So I sang a song, Dolly Parton, Jolene, because that's my daughter's favorite song. Ooh. And I did it. It might not have been the best rendition, But man, was it liberating to stand up there and just belt out a song. And so being able to sort of not take yourself so seriously, get up and sing a song, and seeing these more senior faculty do that, definitely I think is a great thing for the younger trainees to see that we don't take ourselves that seriously and that we can be fun people too. And so I think it was really fun to see everyone get up there and take a stab at a song.
I absolutely love this tradition. I think it's so fun to be able to, like you said, cast aside the sort of austere professional facade that I think a lot of people have when they're sort of awkwardly standing at their poster.
Exactly.
And sort of add this human dimension. I think that's absolutely wonderful. And we talked about a lot of the wonderful work that you're doing in science, but I know there are often barriers. We talked about some of the challenges and struggles, but oftentimes things like funding, staff, technology, feasibility, time, there are so many things that keep you from answering all of the big questions you'd love to tackle. So, Michelle, if we took away the things that normally hold you back and gave you everything you could dream of, what is the one question that you would want to answer most?
It's outside of my wheelhouse. And so I'm assuming that I can take this to becoming an expert in another field.
Oh, yeah.
But the brain really fascinates me, partially because we think of the gut-brain axis, but the brain in and of itself is a very amazing organ to me. And one of the things that I would like to address or solve if I could was dementia, neurodegenerative diseases. It's very difficult to see someone you love go through this. And it may be a sort of selfish thing because I would like to do this because it's so difficult to see someone suffer through that, to sort of lose themselves and lose the person. That it's scary to think of something like that happening to myself, just relying so much of my identity on my thinking and my brain. The idea that someday down the road I could succumb to dementia is actually pretty petrifying. If I had the skills, the training, every resource in the world, I would probably focus my work on understanding the processes that lead to dementia so that neurodegenerative diseases like Alzheimer's would be something that we could actually do something about. Just from a personal standpoint, seeing someone go through that is incredibly challenging. And if I could alleviate that suffering from the individuals and from their families, I think that would just be such an amazing gift.
Absolutely. I think this is such a fascinating area of research, one that I'm particularly interested in as well. And you mentioned, of course, this gut-brain connection, which the more we learn, the more interesting I think the story becomes.
Absolutely. I don't know how many years ago, it wasn't that many years ago that we pretty much ignored the microbiome. We ignored the fact that the gut, which we sometimes call the second brain because of the number of neurons it has in it, that there's clearly connections. We hadn't really thought about that. And so I think that that aspect of science is really coming into its own these days. And I think we will find amazing connections between how things that are processed in the gut, metabolites that are processed, how the bacteria interact with our cells and how the products of that then influence functions, including brain functions. I think there's just so much to be discovered there.
Oh, absolutely. Well, Michelle, I appreciate you sharing your insights and giving me and our listeners out there something to ponder today. And we've covered a lot of ground in our interview today. I'd love to end by talking about advice. So when you think back on your own life, Michelle, your own career, is there one piece of advice that somebody gave you that really helped you that you want to pay forward today?
I think it's a very simple idea. Be kind to yourself. Treat yourself the way you treat your family and friends, with compassion, with grace, with love. We are better people for everyone and everything we do when we are better people for ourselves. So really, we need to speak positively to ourselves and be kind to ourselves when things go well, which is a lot easier, but when things are hard too, we have to allow ourselves to speak positively and just really be kind to ourselves. The other thing I would think about is flexibility. There are many, as I said before, there are many paths to the same destination. So being open-minded to take different paths, I think, is important, and knowing that along the way, our goals may change, and that's okay. We don't have to operate to live up to the expectations of others. We just have to live up to our own expectations and live up to the things that fuel us and motivate us and drive us and that are our passions.
I think that is a wonderful note to end on. I love particularly this comment you made about flexibility in terms of the path that you take in life and the destinations. I think for a lot of people thinking about career paths, some of the destinations that they'll end up don't even exist right now. So the paths are uncertain, and it's okay to not follow kind of that traditional academic research path.
100% agree with that.
Well, Michelle, thank you so much for sharing your insights today. Is there any last note of inspiration or a last piece of advice you personally would like to give our listeners?
Don't be afraid to take risks. Don't be afraid to put yourself out there and take chances, because it's through these moments of being uncomfortable that we grow and that we actually probably make the most impact. So be brave, put yourself out there.
Fantastic message for our listeners. If they would like to learn more about you and the wonderful work you're doing, what is the best way for them to do so?
You can find information about my lab at the Medical College of Wisconsin cell biology page. I am at Twitter at mbattlelab, and my email is mbattle@mcw.edu. So feel free to reach out through any of those means if you want to learn more.
Fantastic. Well, listeners, definitely get connected if you want to learn more about Michelle's work or if you have any questions. Michelle, it was such a pleasure to chat with you today and have you on the show. Thank you so much.
Well, thank you so much for the opportunity. It really was so fun to chat with you today. It's always fun to talk about science and also talk about what makes us tick as scientists.
Absolutely. We've enjoyed getting to know you a little bit better. And listeners, it's been wonderful to have you here with us as well. We hope you'll join us for our next episode of People Behind the Science.